Hey guys, welcome to the video on Chapter 8, Lesson number 2. In this video, I have 3 goals for you guys. I want you guys to be able to summarize the steps in photosynthesis. Second, I want you guys to be able to explain the function of chloroplasts in photosynthesis. And third, I want you guys to be able to describe and diagram the electron transport chain. So, first off guys, photosynthesis, what is it? You've heard it before. You know plants do it. This is a plant thing, so I'll put here P for plant, P-L-A-N-T. And what happens is it takes carbon dioxide, water, and sunlight and turns it into sugar. Now, from a chemical formula standpoint, which you will have to know, so make sure if you ever see this formula, guys, you should have some idea what it is. You are taking 6 molecules of carbon dioxide, 6 molecules of H2O, and turning it into, this here is sugar, oops, wrong button, and you get 6 molecules of oxygen. So basically, photosynthesis is when you take carbon dioxide and water and turn it into sugar and oxygen. Now, it has 2 steps, guys, 2 phases we call them. But these phases, a phase is just the same thing as a step. It would be like trying to play me in a video game. The first step would be to find me. The second step would be to actually grab the video game and have me play it. The first step, guys, is called the light-dependent reactions. The second step is light-independent reactions. Now, if you know your vocabulary from English, you know the word dependent means that you require it. So this means light requiring reactions. This here, though, independent means you don't need it. So here it says light independent reactions. That's a fancy way of saying you don't need to have light. So hopefully that makes the 2 steps self-explanatory. The light-dependent reactions require light. The light-independent reactions don't require light. And you will need to know which one of these comes in order, and we will be going over that here in a minute. So the very first step, guys, the very first step that you will see, so I'll put here first step, is the light-dependent reactions. These all happen inside of the chloroplast. Now, if you remember from chapter 7, we covered a little bit about this inside the cell. The chloroplast is an organelle within a plant that is responsible for photosynthesis. Its one job is to make photosynthesis take place. Now, the chloroplast has 3 parts to it that you will have to know about. The first one is called the thylakoid. The thylakoid are these little pancake-shaped looking things here on the right. And these are where the light-dependent reactions take place. So when I say there are light-dependent reactions, I am saying that they are happening here inside of these little disc-looking objects. These are called thylakoids. Now, a collection of these things, a collection of thylakoids is called a granium. So, when you see a collection of these thylakoids, like you see right here, or even right here, or here, or here, a collection of these is called a granium. And you'll have to know, if I ever mention the word granium, you need to know that I mean that it's a stack of thylakoids. Now finally, there is the stroma. The stroma is basically located in between. You see how there's lots of open space here, guys, inside of the chloroplast where there's no thylakoids at all? That stuff right here is the stroma. This is just the fluid-filled space outside of the thylakoids. This is where the light-independent reactions take place. So, the phase 1, the light-dependent reactions take place inside of the thylakoids, and a collection of thylakoids is called a granium. Now, this chart right here is the bane of existence of biology students around the world because it's difficult to understand. So I'm going to walk you guys through it step by step and hopefully get you guys to understand what's going on. This here, guys, is a picture inside of a thylakoid. So this part here is the inside of the thylakoid. This part here is the stroma, which is outside of the thylakoid. What happens is this. These things right here, pictured, the green things, these are all proteins. They're all enzymes. Their job is to make chemical reactions take place. And if you remember, guys, the role of enzymes, I-M-E-S, is to speed up chemical reactions. I'll put RXN for reactions. So all of these things here you see in the membrane. The membrane is this blue part right here. If you don't remember this little picture from all the pictures of cells, these things right here are all enzymes, and they're going to help reactions go faster. So the very first enzyme takes in sunlight, and that takes that sunlight and it breaks apart a molecule of water and releases two hydrogens and releases an oxygen. And then what happens is that makes this side of the membrane down here have a lot of hydrogens. Because if you see here, right here, it's releasing two hydrogens. Well, that's going to give the inside of the membrane, as you see, a lot of hydrogens will be floating around. On top of that, what happens is that these proteins right here are going to grab hydrogens from the outside, inside of the stroma, and they're going to bring it into the thylakoid space. So the very first job, guys, of the photosystem 2 is really to bring hydrogens into the thylakoid space. And that will make more sense here in a minute. So the first job here, guys, is to get more hydrogens. And that's the job that happens at this first step right here. Now, I haven't mentioned up right here yet. Right here, guys, there's an electron. Electron, if you remember from chemistry, is the thing that can float around an atom. And right now, when you split the water, it created a free electron that can float around. This electron is going to follow the path going to the next step right here. Now, right here, the electron is used, in this part right here, the electron was used to bring in an extra hydrogen, which is good. Right here, what happens is that that's going to use energy though. When you're going to bring in a hydrogen, you're going to lose some energy in the process. The role of these next two proteins is to basically re-energize the electron. It gives the electron its energy back. So here you can think of it as having low energy. Then light comes back in, and this enzyme gives that electron more energy. At which point in time, it travels through these proteins right here. And you can think of these proteins here, guys, as just a road. They're just giving the electron something to travel through. Until it reaches this very last protein right here that we called ferrodoxin. And what ferrodoxin does is it grabs that electron, takes a molecule we call NADP plus a molecule of hydrogen, and creates a molecule called NADPH. NADPH I will discuss later on, guys. But understand this is a very, very important molecule. NADPH is essential to photosynthesis, and it's going to be coming together later on in the light independent reactions. So understand, at this point in time, guys, electron is done. It's now part of this molecule here, and it's finished. But let's go back for a second. We have all these hydrogens down here. Why do we have these hydrogens? Why does the process of electron transport want to bring these hydrogens into the thylakoid space down here? Well, the question comes down to is right here. The answer is right there. An enzyme we call ATP synthase. Its job is to take hydrogens and attach those hydrogens to a molecule we call adenosine diphosphate. That might sound familiar. If you remember, there was a molecule from chapter 8, section 1 called ATP. Well, ADP is just ATP, except it's missing a phosphate. So ATP synthase takes an atom of hydrogen, and it uses that combined with a phosphate, which isn't labeled here, but I'm going to draw it in there anyway. and makes ATP. So what you've done here, guys, is you've taken water and sunlight and you've changed it into NADPH and ATP. These two molecules here, guys, are going to move into the next step, which is called the light independent reactions. And that's electron transport. This diagram here, guys, will take a lot of time for you to understand. You will need to go over this video probably a couple of times to get it. So if you understood this first time, awesome. If you didn't, don't be surprised. It's complicated and it's difficult. Focus on what the goal is. The goal is to make NADPH and ATP. And then focus on after that, you should ask yourself, okay, how do you do that? Well, you need sunlight and you need water. Everything else is just steps in the process to get there. So, a quick little side venture. Pigments. Before back here, guys, you might have noticed that we've kind of shown these things as being green, and that's because we typically think of plants as green. Well, plants use chemicals we call pigments. Pigments are just light-absorbing molecules that are found inside of thylakoids. Now, there are three types of pigments. There's chlorophyll A, chlorophyll B, and carotenoids. Each one can absorb a different wavelength of light. If you see right here, guys, this chlorophyll B typically absorbs blue the most, which means that it's going to look more red. It's going to look more green because green is where it's at the bottom right here. Chlorophyll A absorbs mostly dark blue and purple with a little bit of orange and red. Carotenoids, on the other hand, they absorb light blue and some orange and yellow. Plants are going to have a combination of these chlorophylls that are going to let them absorb the light. So when I say absorb the light, if you see right here how light's coming in and affecting this enzyme right here, this right here is a type of chlorophyll. It's going to be absorbing the light. And there are three different types. So when I mention pigments, guys, I'm talking about molecules that can absorb light, and they absorb light to help make photosynthesis actually happen. Okay, next step, guys. Phase 2. So at the end of the electron transport, we have two molecules. We have NADPH and ATP. Both of these are high energy molecules. They have lots of energy in them, but there's a problem. They break apart very quickly. It would be like having a piece of very breakable glass. It looks cool and it works, but it breaks way too easily. You don't want it to break. You don't want your ATP and NADPH to break. Now, for those of you guys who are curious, I say, what do you mean by break? What I mean by breaking is you don't want them to return back to their old steps. You don't want ATP to return to ADP. That would be a bad thing and it would hurt you. Same thing is true with NADPH. You don't want it to break it. So in order to solve this problem, you have to have some kind of a stable molecule that can store energy while it's needed. And that molecule is called glucose. And glucose is a sugar. And what happens is that in phase 2, the light independent reactions, the Calvin cycle takes place. The Calvin cycle is turning of carbon dioxide into a molecule called C3P, which is then used by using ATP and NADPH as a source. Well, two C3P molecules can be turned into glucose. So the whole purpose of the Calvin cycle, guys, is to create a molecule called C3P. The Calvin cycle is powered by NADPH and ATP. So let's take a look. This here is a diagram, guys, of the Calvin cycle. What happens is that you get 6 molecules of carbon dioxide. You then convert that into a molecule called PGA3. Now, you don't have to know the names of these molecules. At most, guys, I will require you to be able to draw this out. But the names here, I don't too much care about. So what happens is that 6 carbon dioxide is then used with 12 ATP and 12 NADPH, and that creates a molecule called G3P, which you might remember from the word back here. I mentioned it back in the previous part. Actually, no, I didn't. Sorry. Oops. Never mind. The G3P comes here. Oops. My bad. So what then happens then is you get G3P, then gets turned into a molecule that looks like this, 6-ribose phosphate. It's basically a 5-carbon molecule that does its thing. At which point in time, it kind of split apart. What happened here is some of the carbons went this way and some went that way. Some of them were turned into a molecule called 2G3P. G3P is going to later on going to be turned into glucose. This molecule right here, though, is going to continue on. It's going to be combined with 6 ATP to give it some of its energy back, because this is a low energy molecule. This here is a high energy molecule. At which point in time, you're going to get 6 more carbon dioxide coming in, and the cycle is going to repeat itself. This cycle, guys, is confusing. There's no way around it. You are going to have to watch this part of the video a couple of times in order to fully get it. And I have a TED Ed video as well you can watch as well. Understand here, though, the goal is this. The goal is to take 6 carbon dioxide, combine it with ATP, NADPH, and to make glucose. That's all the Calvin cycle does. So, before I'm done, guys, I have 2 more quick things. Now, there are 2 alternative ways plants can do this, and I will only briefly talk about these, and I will probably only require that you have some memorization of these ideas. So, the first one is a C4. Some plants make a molecule with 4 carbons rather than 3. So, if you look back here, you see how I'm making a molecule with 3 carbons? The C4 pathway instead makes a molecule with 4 of them. Plants in dry climates do this because making this C4 molecule helps them to save water. You don't need to know how it helps them save water. You just need to know that it does help them save water. The other one is a type of plant called a CAM plant. These are plants that save water by only letting in carbon dioxide at night. That prevents water from escaping. Think about that. If you are letting in carbon dioxide during the daytime, well, daytime tends to be very hot, and that means you're going to lose more water. Just E on that one. So these plants decide, okay, well, let's skip that problem. Let's just open up at night when it's cold, and that way I won't lose as much water. So these plants here, the CAM plants, what they do is they run their light dependent reactions during the daytime when there's sunlight, and they run their light independent reactions at night. This saves them the trouble of having to lose water during the daytime to let in carbon dioxide. It's a cool little trick, and I would probably say all of the plants here in Saudi Arabia are at least one of these 2 types, because obviously it's hot here in the kingdom and you don't want to lose water when you are a plant. Okay, guys, I am basically done. If you need some more help, guys, with the Calvin cycle, I have posted a video online that you can use on the learning management system that will give you kind of an animated vision of what the Calvin cycle is. So I would highly recommend going to go watch this video at some point in time. It does a more animated way of explaining what I've already told you. It's the same information I've told you, it just looks cooler and might maybe be easier for you to understand. That's it, guys. This part right here is just lesson review. It's all the key terms from the chapter you are going to need to know. And I am done talking. I hope that you walked away from this with a better understanding of photosynthesis and how it works. And thank you very much for sticking to the end, and I will see you guys later.